Yes, class. Am I audible?
Yes.
So, Akifa, Atif, Fatima, Hamza, Hanifa, Hiba, Sarah, and who is this? Zen. So, class, we'll start with optical instruments. Rest of the part of lens formula we had already completed in the last class. Now, optical instruments is a topic that is left, and one more topic that is left is prism. So see, optical instruments are what? Optical instrument means any device that helps you to expand your vision, whatever is your range of vision. If you are able to see up till this much distance, then it can increase it or it can decrease it up according to your need. Now, even we say our human eye, under this category of optical instruments, we have eye as well, because not only the devices that help you to extend your range of vision, it is these also include the devices that help you to see. So eyes are the basic human devices that help us to see. That's why eye will definitely be included. Then in your instruments, you have a microscope and a telescope. Now, I is not included in your syllabus. See, optical instruments questions do come in your exam, and maximum chances are there questions will come from optical instruments. Last year also questions came, so chances are there. So, I is not included in optical instruments now. Means you will be able to find it in NCERT, but in NCERT it is given hypermetropia and all the diseases are also mentioned, but now questions are not asked on this, so that's not a part of your syllabus, so no need to go into the details of eye. Quick revision of eye we'll do. Eye is basically what? Let me tell you whatever is important for you. Eye, your human eye outermost layer has cornea, then it has sclera and choroid membrane, all these three layers. Cornea is the outermost membrane. Then after it, you have iris. Iris is a colored structure which provides the color to your eyes. Whatever is the color of your eyes, that is given by the iris only. And then you have pair of ciliary muscles, and these ciliary muscles help to hold the lens. And they contract and relax, and based upon their contraction and relaxation, the accommodation of lens occur. So whatever is the requirement of the lens, that power of accommodation is done by the ciliary muscles. Then in between the cornea and lens, you have a chamber, and between the lens and the last boundary or wall, you can say, you have a chamber. This is aqueous chamber, this is vitreous chamber, in which you have fluid. Fluid is always known in terms of humor. You say fluid whenever you even refer to blood or any liquid, you refer to it as humor. So you have aqueous humor, you have vitreous humor, and in the retina, this is the final wall where image is actually formed, and this image is inverted. Here, the image that is formed is inverted. So optic, there are many centers present over here. You have fovea also, you have blind spot also. One of the areas has optic nerve. Optic nerve takes up the image, inverted image that has been formed, and it takes it to the cerebral cortex, and wherever you have visual motor area for sensation, that time it is recognized. Like this, the simple working is there. Now you don't need to go into the details of this. Now you don't even need to go in. You just have to remember eye lens is a convex lens, so wherever human eye will be there, human eye will be referring it in terms of lens only. So this is about eye. Then you have microscope. Microscope we'll study today. Both the types of microscope we'll cover. There are two types of microscope basically. One is a simple microscope, other one is a compound microscope. Telescope, let's talk about it later. Now, 2 points are there which are very important before we start with any of the things. See, near point and far point, because we will be dealing with everything in terms of near point and far point. Near point is the closest distance from your human eye, from the human eyes, in which you are able to see an object clearly. For example, if I keep this pen very close to my eyes and I try to see it, I'm able to see two images or it's very blur. I'm not able to see it. If I'm keeping it as a distance, this much distance, still I'm not able to see it. It is becoming blur and it is giving strain to my eyes. But as if I'm keeping it at this distance, I'm easily, clearly able to see one single pen's images. There's no strain that is put on my eyes. So it means this is the closest possible distance in which I am able to see anything. This distance is actually 25 centimeters. And this is also known as least distance of distinct vision. Least distance of distinct vision. It means this is the minimum distance in which you are able to see an object clearly. Now, what is then the maximum distance in which you are able to see an object? That is known as the far point. Means the farthest distance that you are able to see from the human eyes. Farthest possible distance. So, farthest possible distance is infinity. See, anything that is separated to you, you are able to view it. You are able to see it. Large mountains are there, from a very distant place you are able to observe it, or even you are able to look at the sun. It's a different thing that size is smaller and everything, but image of the sun, or if object is sun, the image is formed in your retina clearly. You are able to observe moon, you are able to observe the stars. They are at such a huge distance from us where we are right now, and the place they are residing, they are separated by light years, but still you are able to see it, because far point is infinity. Till wherever your human eyes can see, that point is infinity. So you say far point, this is infinity. Far point is infinity. Now see, as I was saying, we'll start with microscope, simple and compound microscopes we'll do. But one thing is here, in both of these simple and compound microscopes, we'll see two cases. First, we'll see when the final image will be formed at this least distance of distinct vision, that is 25 centimeters. And one case we'll see when the final image is formed at infinity. Both the cases, near point and far point. Similarly, for compound microscope also we'll see least distance of distinct vision and when the image is formed at infinity. Both these cases we'll study. So this we'll study, we'll start with simple microscope directly. But before that, just copy this down, then I'll start with simple microscope. All right, let's start with simple microscope. So see, for simple microscope, one single convex lens will be used. One single. When you will reach to compound microscope, you'll be having two lenses. So that's why understand the case of simple microscope clearly. Simple microscope, a simple convex lens will be used, which will be having a small focal length to magnify object that has been written. So as I've told you, we'll be dealing with two cases. When your final image is formed at least distance of distinct vision, and one case when your final image is formed at infinity. Fine. Now see, why are we using microscope? All of you must have used microscope once, at least in 9th or 10th standard. All those who do not have biology right now, they must have used it in their earlier classes. And those who have it with them, they must be using it right now only. So simple microscope, if you want to enlarge image, you want to magnify image, you use simple microscope. Compound microscope, if you want to magnify the image by 1,000 times, if you want to magnify the image by a greater number, then you use compound. Right now we are dealing with simple microscope. Fine, it is used to magnify image. Now see, if there is no, let's say, if there is no microscope, object is there and the human eye is there. This is the object AB, right? If no lens is present, then what will happen to the rays? Rays will simply direct, it will reach the human eyes without any refraction, any deviation. Like this, here you will be able to see the way this is an object. There is no presence. Okay, there are specks right now. Now you can say there are no presence of any kinds of lens in between. Now what will happen? I have this pen with me. Now if I look at this pen, if I'm looking at this pen, there is no presence of any lens to refract it. If I'm looking at it directly, it will be subtending an angle at my eyes. So that is what is written over here. Let's say this angle is alpha. Okay, so this is the angle subtended by the object on eye. So can you tell me how much distance this should be? Let's say this is human eye. Can anyone guess it? How much distance this should be? I'm able to see this object clearly at this point. This distance should be, any idea?
25 centimeter.
25 centimeter. Good, Fatima. It should be 25 centimeter. That is least distance of distinct vision. The reason, if I'm able to see this clearly, it means it is at my least distance of distinct vision. Fine. Now coming to the lens part. Let's say this is the object AB. Fine. So accordingly, the rules of ray diagram will be followed. One ray will go. This is just to make it in continuity. On the screen, it is difficult to draw it. So ray got refracted over here. Fine, this is the optical center and the ray of light got refracted. And the second ray the way the second ray has to pass through the optical center, the way the ray has to pass through the radius of curvature. Similarly, here the rays have to pass through where? Here the rays have to pass through the optical center. Fine. ray has refracted. Why have I tilted it? Because otherwise the ray diagram would have gone a lot and the screen will not be enough for it. That's why I've shifted the angle of refraction slightly. So if I extend it at this point, so rays are appearing to meet at this point. This becomes A dash, B dash. Can you see a magnified image has been formed now? So now where do you keep your eyes when you observe this? Whenever you see a magnified image, you keep your eyes over here. When you are placing your eyes in that binocularish space in the microscope, eyepiece which you say, there you are actually placing your eyes over here, from where here you are able to see. Object is kept at a place where we are not able to observe all the slides and everything that is present. Fine. Now see, one thing here, this angle that is subtended by this object, this image on the eye. Can you see this image also subtends an angle? This angle here. Let's say this angle is beta. Fine. This angle is beta. Now what we have done? Final image distance. This distance should be how much? We have already taken the case when the final image is formed at least distance of distinct vision. So if final image is formed at least distance of distinct vision, this is my final image. So this distance should be capital D. And remember, eye and lens are very close to each other. Eye and lens are very close to each other. Fine. So do not consider and worry about this separation. This is actually very small. From here only you are able to see it. Till here, is it clear? Yes or no? Fatima, Hanifa, Hamza.
Yes.
Okay. All right. Now see class. If you have understood till here, no. Now angular magnification is very easy. Angular magnification is simply the ratio of what? The angle that is subtended by object on eye when it is at least distance of distinct vision. So this is the angle that is subtended by the object on eye when it is at least distance of distinct vision. To the image's angle that is subtended, which is when image is at least distance of distinct vision. So angle subtended by object on eye and angle subtended by image on eye. So it is actually image by object. So here what you will have the ratio. It is angle subtended by the image on the eye, beta, and the angle subtended by object on the human eye, that is alpha. Why are we considering this, not this? Because we have to have this least distance of distinct vision. Now see, okay, I think I've written it already. It is the ratio of
Alpha.
Alpha is this. This is alpha. Alpha is the angle that is subtended by an object on eye when the object is at least distance of distinct vision. Means like this, simply like this, without any lens. If I'm viewing this, the angle that this object is subtending at my eyes, this is alpha. Clear?
Yes.
Okay. Now see, so magnification will be what? It is the ratio of angles subtended by image and object on eye when both image and object are at least distance of distinct vision. Both were at separation of D, capital D, and it's the ratio of angles subtended by image, so beta divided by alpha. Now, if you look at this triangle, look at this triangle for tan alpha. This triangle. If you write tan alpha, and I've told you small angle approximation you can use because the angle that is subtended, that will be subtended at the human eye will be very small. Obviously, in the retina, how much space is available for the angle to get subtended? Very small angle will be formed. So here, angle that is subtended by eye is alpha. So here you can use small angle approximation. In small angle approximation, you can write tan alpha is alpha, tan beta is beta, tan theta is theta in short. So here in this triangle, tan alpha is AB divided by BE, right? Perpendicular divided by the base, AB divided by BE. Fine, that is tan alpha. Regarding tan beta, if you look at this triangle, A, B, O. So tan beta will be equal to AB divided by BO, right? So I'll use these only. So in triangle ABE, tan alpha, which is almost equal to alpha, is equal to AB divided by BE. In triangle ABO, tan beta, which is almost equal to beta, is equal to AB divided by BO. Now what can I do? I can keep the values of alpha and beta here. So beta is AB by BO. Alpha is in the denominator, so I am reciprocating it, divided by AB. AB gets cancelled. Magnification becomes equal to EB divided by BO. Now see what is EB and what is BO. EB is already, as Fatima answered, this is least distance of distinct vision, capital D. Now, where is the lens over here? Why are we referring it? Because lenses are there in human eye. So how will you, for choosing the sign convention, no actual artificial lens is kept here. So how will we choose the sign convention if no lens is there? Object distance, how can we take? So you can refer it from your human eyes. If this object is in front of the lens, that is in front of human eyes, this distance will be negative, minus D, minus capital D. And second distance we had OB. See, this is OB. So this is OB is basically object distance. That becomes minus U. Clear? So EB is minus D, OB is minus U. Fine. Now, if I write the formula of magnification from here, clearly magnification will be equal to D by U. But see, using this formula becomes a little difficult. So what we'll do, we'll use the lens formula that we have studied. What was lens formula? Lens formula was simply 1 by F minus 1 is equal to 1 by V minus 1 by U. From here, C class, focus and final image distance we have to write. Object distance is minus U only. Image, final image is formed where? This is the case that we are taking. Final image is formed at least distance of distinct vision only. Right? Both the angles, both the object and images were at least distance of distinct vision here. So image distance is minus capital D. Look here. Final image was here. This is O. And we have chosen the case where this is actually equal to minus D. And focal length for a convex lens, that will be positive F. If you put it in the formula, you get minus 1 by D plus 1 by U is equal to 1 by F. From here, you can write 1 by U as 1 by F plus 1 by D. Now, coming to this formula, M is equal to D, small d, capital D. So this becomes capital D multiplied by 1 by F plus 1 by capital D. See, if you multiply it within the bracket, what do you get? Capital D divided by F plus D by D is 1 only. Get M is equal to DF plus 1. This is the formula for angular magnification. Now tell me, is it clear or not? angular magnification part. So final formula you have to remember, D by F plus 1. And one more thing, class, here you don't need to use any sign convention. In optical instruments, you are free to write whatever is the magnitude. No need to write the sign convention. I'll write it only. No sign convention. No sign convention. Fine. Noted till here. Any queries you have in between, ask me.
Ma'am, can you show the previous notes for a moment?
This one.
Yes, ma'am, just for a moment.
Okay.
Okay, ma'am, thank you.
Fatima, your mic is on.
Ma'am, can you scroll up for a moment?
So by now you must have written it. Anyone is writing, stop me, I'll scroll back. Otherwise, see case 2. So I'll modify here only. You all have to draw it again in your notebook. Otherwise, it will become very confusing for you. Final image is formed at infinity. That will be your case 2. Okay, make this heading. Now see here what will be the difference. Object angle subtended by object will still be alpha, because even if the final image is formed at infinity, final image is formed at anywhere. If I keep the object like this, at least distance of distinct visual will remain the same always. So this case will remain the same. But in the ray diagram, you will not be having image formed at least distance of distinct vision. This will not be there. See here you have the object AB. One ray will pass through the optical center. Other ray will go and get refracted. But see, as you will produce this, these image will not appear to meet anywhere. They'll meet at infinity only, because your final image is formed at infinity. Fine. So since the final image has been formed at infinity, now your least distance of distinct vision distance has changed. Fine. This is object distance. Final image is formed at infinity. This is the change that will occur. Now certain things will change. What will be the change that will come here? See, magnification's definition will remain the same. It is still m is equal to beta by alpha. From triangle ABE, you can get alpha as AB by E. In triangle ABO, you can get it again as AB by BO. Until here, you will get it back. Now see, difference will come here. Difference that will arise will be from this part. No need to write all this. See class, the difference that will come in angular magnification will be object distance. Here, if I'm referring to the separation OB, see, if image is formed at infinity, then where according to you object would have been kept? If your final image is formed at infinity, then where have you kept the object that you're obtaining the image at infinity?
The focus.
Focus. Yes, both of you are right, Atif and Sarah. Image is formed at focus. So it means this is kept at focus. So this distance is now focal length. So instead of you will be using F. So this becomes minus F. This becomes F. Formula becomes D by F only. In case of least distance of distinct vision, you have D by F plus 1. Here in infinity, you just have D by F. Quickly note it down, then we'll do a numerical on this. Make it like this. Case 2, final image is formed at infinity. Repeat it because in your notes, you should have it written. Again, no need to use sign convention. Even if you think the focus is formed in front, if you think the focus of whichever case you have is in front of the lens, still just write the magnitude. No need to put the sign convention. Done. Image formed at infinity, angular magnification we have done. Okay, it says a converging lens of focal length 6.25 centimeters. So focal length is given as 2.4 centimeters, 6.25 centimeters. This is used as a magnifying glass if near point of observer is 25 centimeters. So in some questions, 25 centimeter can be mentioned, but there are chances that 25 centimeter you have to write on your own. So just in case in questions where you do not have any information about the near point, you have to use 25 centimeters only. Calculate distance of object from lens. So distance of the object from lens is basically object distance. It is asking you to calculate, right? Distance of object from the lens means object distance. Focus is given. Image is formed at least distance of distinct vision, right? 25 centimeters is given. If 25 centimeters is the near point, it means image is formed at that point. Angular magnification when image is at infinity. So see, focus is 6.25. So for the first part, we'll use the sign convention because we have to use the lens formula. And in the lens formula, we use sign convention. Here in magnification and this optical instruments part, we don't use it, no. So see, 1 by f is equal to 1 by v minus 1 by u. From here, you just have to find out u. U is unknown. So see, if you calculate 1 by u will be equal to 1 by v minus 1 by f. So this becomes 1 by u will be equal to 1 by minus 25. Both are in centimeters, no? So no need to convert anything. Minus 1 by 6.25. So you get 1 by u as what? Minus 1 by 25. Minus 10 here is there. 66.25 will become, if you remove the 0, 1. 100 will become 625 and 25 LCM. So 30 is by 2, 15. It becomes minus 25 minus 10 by 625, no? So this should be minus 15 centimeter. Somebody please cross check whether it's minus 15 or not. I have done it orally. That's a mistake. Now angular magnification. So this way, using lens formula, you will calculate angular magnification when image is formed at least distance of distinct vision is d by f plus 1. So we have d as 6.25. No, sorry. D is how much? 625. Focus is this. D is 25. Focus is 6.25 plus 1. So this becomes 4 plus 1, 5, right? And third part is angular magnification when image is formed at infinity, means only d by f. So that is 25 by 6.25. That will give you 4. So C class, just a small conclusion of what we have studied. We have studied simple microscope. In this, we have studied 2 cases of it. First case is when image is formed at least distance of distinct vision, and another case when image is formed at infinity. This is the final image. Also, in this, we have seen magnification's formula is d by f plus 1. Here, magnification's formula is only d by f. Till now, we have studied this part. Now, we'll start with compound microscope, but note this question, then we'll start with compound microscope.
Teacher.
Yes,
Teacher.
Fatma. Yes, Fatma.
Yeah, we are outside, so can I leave now? I will look at watch the recording
Okay,
afterwards.
okay. Fine, Fatma.
Okay, Allah Hafiz. Thank you.
Completed? Let's start with compound microscope then. Zen, do send your question paper as well to me. Fine, the question paper you have in your exam. Okay, class. So see, compound in microscope, no, the difference is just that two convex lens will be there. One convex lens will be having a shorter focal length, shorter aperture, that will be called as the objective lens. It will be kept near the object. And second lens will be eye lens. Eye lens will be kept near the human eye. It means the lens into which you will look into, that will be the eye lens. And it will have a comparatively larger aperture as compared if you compare it with the objective one. And compound microscope, since two lenses are there, so refraction occurs two times, more than two times. So you can say it can magnify image 1,000 times. So it magnifies images 1,000 times. So that's what has been written on the screen. Compound microscope, it consists of two convex lenses of short focal length and short aperture. It can magnify 1,000 times. Construction involves one objective lens. It is positioned near the object to be magnified. It is convex lens of very short focal length and short aperture. While the eye lens, it is positioned near the eye to view the image. It is convex lens of comparatively large focal length and larger aperture. Fine. Now see again, two cases we will see when the final image will be formed at least distance of distinct vision, when the final image will be formed at infinity. Now see, firstly, again, the same thing we'll do. If object is there, let's say object is there, you will view it without the lens first. If you view it without the lens, then at your eyes, it will get, the angle will be subtended. Human eyes will be able to see. So this angle is capital D. Now this distance is capital D, which is least distance of distinct vision. Now coming to your lens, proper lens. So two lenses will be there. Now see, first lens, drawing the first lens is not difficult. It's easier because first lens for you, it acts as a normal lens that you have studied till now. Like if I am not drawing the eyepiece right now, let's say this is the objective lens. This is the objective lens, fine. If I keep the object like this, I have kept the object, let's say this is AB. So I have not drawn the eyepiece yet. Now see, what will be the path of the rays? Firstly, it will pass straight away. It will get refracted. Right? It will get refracted. And second will pass through the optical center. So at this point, it is appearing to meet. So this point becomes A dash, B dash, right? Now see, so was there any difference still here? Any new concept here? No. Object one lens is there. An object is kept in front of you. An image has been formed, right? Now see, this is since this is objective lens. This separations is u naught, mean separation object distance of objective lens. Let's say this is some point focus is there where light rays have converged. So this becomes the focus of the objective lens. Fine, and since the image is formed here, so this separation becomes the image distance. Clear? Now coming to the compound microscope actual part, which involves your lens as well. So you will have a bigger lens, which will be referred to as the eye piece. You can say eye lens is also said. Draw it properly. I have not drawn it. Now see what will happen to the rays. These set of rays, these will get refracted actually, but it will become difficult to show it, so I'm drawing it directly. Now see, let's say this is A double dash, this is B double dash, fine. So here the human eyes will be there in front of above the principal axis. So basically what happened, this is also actually joined. This also got refracted. This is within the lens only. The rays, this intermediate image acts as what? It acts as the object distance for this lens. For the eyepiece, this acts as an object, virtual object, only a virtual object, not a real object, but this acts as a virtual object, this intermediate image. So light rays, this intersection point image should be here. This is actually the intersection point. This is A dash, B dash, not this. Where the intersection is occurring, this is the image distance. Now what is happening? Now rays get refracted here. Rays get refracted. And if you produce it, you find an image over here. All right. Again, angle subtended by image on the eye. This has to be straight. Otherwise, just a second. Let me rewrite. Now it's fine. Just shift the eye over here. At this point, you will be having the human eye. This is just kept above. So at this point, you will get beta, angle subtended by image on the eye. Now see, when you will study magnification, definition will still remain the same. Definition will still be tan alpha, that is alpha divided by beta. So see here how the difference will arise. Magnification is beta by alpha, and tan alpha will still be the same. Tan alpha will be equal to AB by BE from triangle ABE. Now in tan beta, you'll have a different triangle now. See, tan beta will be this A dash B dash. Actually, this has to be a proper triangle. B dash A dash. Yes. Let's take this point as what? O2, optical center for the second one. O2, take it as O2. Now, see, if you look at this smaller triangle, A dash, B dash, O2, here you can say A dash, B dash divided by B dash, O2 will give you tan beta. Tan beta will be what? Perpendicular divided by the base. So from here, if you have it, in triangle A dash, B dash, O2, tan beta will almost be equal to beta. It will be equal to A B dash, A dash, B dash divided by O2 B dash. Now see one more thing. A dash B dash is what? If you see it properly, A dash B dash is actually height of image. A dash B dash is the height of image, and O2 B dash is the object distance for the eyepiece, for the eye lens. So you can directly say AB divided by. This part is what? This is okay. Let's take the ratio first. Let's take the ratio. Beta by alpha. So beta here is A dash B dash divided by O2 B dash. Alpha is BE divided by AB. Fine. This also you can write. Now see here you have the proper magnification formula. Tan alpha is already there with you. Now I was saying, yes, I was saying that here if you see A dash B dash is actually the height of image that is formed, and O2 B dash, if you look here, A dash B dash is height of image that is formed, and O2 B dash is the object distance for the eye lens, UE. Right. So this I was saying, we can write it as tan beta can be written as, or here only I'll write A dash B dash can be written as height of image. O2 B dash can be written as object distance with a negative sign. Now, what are AB and BE? This also we have to write about AB and BE. AB is the height of object, and BE, this point E. BE is what? This is the least distance of distinct vision. So BE becomes minus D, AB becomes height of object. From here, anything gets cancelled? No, only minus sign gets cancelled. This is one thing. Magnification's formula is height of image into least distance of distinct vision divided by height of object into UE, that is object distance for the eyepiece. Now see, we'll still not be using this. This formula will not be used anyway. This is of no use to you. But if you see height of image by height of object, if you look at this carefully, this is actually the magnification of the objective lens. Objective lens was a simple normal convex lens for you, remember? So if it is a proper normal convex lens for you, then what is convex lens formula of magnification? In the last class only we did it, that it is simply height of image by height of object. So height of image by height of object is magnification of the objective lens. All right. So and magnification of objective lens, you can even say this is V0 divided by U0 as well. Both of these things you can say. Now, main question is, and height of eyepiece. Eyepiece is a proper simple microscope for you. If you forget convex lens, eyepiece is actually acting as a simple microscope if you do not consider the first lens. See, if you hide the second eyepiece, if you hide it, you get a, I'll show you the figure, what I'm trying to tell you. If you hide this eyepiece that you're getting, for you, this acts as a normal convex lens whose magnification will be height of image by height of object or V0 by U0. If you forget this objective, if you just remember this is here, then for you it will appear as a simple microscope, which is enlarging the image. So if it is acting as a simple microscope, which is enlarging the image, so magnification will be capital D by F plus 1. Fine. Now magnification is MOME. This is the combined magnification of both of these, which you have studied. So magnification is what? This is minus sign will come here. So magnification will be, because height of image is negative now here. So minus V naught by U naught is there, and instead of ME, you will have D plus F, D by F plus 1. Now one approximation is there which you have to remember, that is C class. Sometimes no, the length of tube is written, which is equal to V naught. Length of tube means this. This is the length of tube. Separation between the eye lens and the objective lens, that is known as the length of tube. So sometimes length of tube, instead of writing this complete, this is written as V naught only. All right, so V naught is written as capital L. V naught is sometimes written as capital L only. And you write focus as, you write objective lens as the focus. If you write the objective distance for the object lens, object distance for the objective lens, that is written as the focus. So formula just changes, minus L by F, D by F plus 1. Now difference, this is FE. Here also it was FE only. Fine, because you're referring to eyepiece here. This becomes F. This is the difference that arrives. So main formula is this. Modification of that formula can also occur in this. Note it down. So second case is when final image is formed at infinity. So now what will happen? Everything here will be the same. Final image will be formed at infinity. This will be the second case. So here the human eyes will be there. And final image will be formed at infinity. And this becomes beta. Now see, if final image is formed at infinity, then again here UE will be replaced by FE, same thing. This will be the focus, no only, then only final image is formed at infinity. So again in your, this part also till here it will be the same. Difference will arise in UE. HE, here you will have this as UE. UE will become FE. So this becomes FE, this becomes FE. Fine. Now in this formula, here also, for the magnifications of lens, for final image at infinity, you have just D by FE. So this term will not be there. And this becomes the final formula. Again, no need to use sign convention here as well. All right, note this down again. Make it as case 2. Make this as case 2 when final image is formed at infinity. Also, one more thing, class. If this is L, no length of tube, then this is actually V naught plus UE. Right? This is the complete length of tube, which is comprised of V naught plus FE. FE or UE. Yes, class, just text me done once you have completed it. I'll stop the class then. One numerical we'll do on compound microscope in the next class now.
Done, ma'am. Thank you.
Okay, all those who have completed, you all can leave now. Thank you so much, class.